ஸ்ட்ரீம் இது கூட கொடுத்துருங்க மேம். ஸ்டார்ட் ரெக்கார்டிங் கூட.
உங்க கேமரா ஆன்ல இருக்கு மேம். உங்க ஃபோன் யாரையாவது அந்த யூடியூப் லிங்க்ல ஆகும் மேம். செக் பண்ணுங்க. என்ன வருது மேம்?
வந்துருச்சு மேம். ரெக்கார்டிங் ஆன் பண்ணிட்டேன். ரெக்கார்டும் ஆகும் மேம். ஸ்டார்ட்
அதான் யூடியூப்ல இருக்கேன் மேம்.
ஸ்டார்ட் ரெக்கார்டிங்.
சரி மேம்.
இப்போ நான் இதை மட்டும் ஆன் பண்ணி விட்டுருங்க. I should minimize this. லைவ் ஸ்ட்ரீமிங் மட்டும் ஆன்ல வச்சிருக்கேன். இந்த
selectively and minimize the unwanted radiation to the other healthy tissues. In that case, we can achieve more target-based treatment as well as target-based diagnostic purpose. Like this slide gives the beauty of the target specific radiopharmaceutical, like the fluorine 18. It's called like it is labeled with the fluorine 18 with the fluorodeoxyglucose. Like this is routinely commonly used for the diagnosis of cancer. Like how it does it work mean, like depends upon like during cancer progression, there are several glucose metabolism pathways involved. This tracer selectively accumulate on the glucose uptake. That's why we can visualize the tumor. But this patient is suffering from adrenal tumor. They treated with this this is non-target. Like it's not target based. It's based on the glucose metabolism. It's used as a diagnostic agent. When they treated with this patient with the FTG, they failed to identify the cancer. But the same patient, when they labeled with the zirconium-89 with serentuzumab, that is their target-specific agent, injected to patient and find out the cancer location on the adrenal gland. Like it's very early stage, the patient is identified by this specific tracer due to the target selectivity. Again, the patient no need to go for the classical chemotherapy for the treatment. They can go for selective chemotherapy for this patient. It gives the precision and personalized medicine treatment option too. Like again, I said we can use this tracer for the radiotherapy. In radio, if you want to use this tracer for radiotherapy, we have to label with the different isotopes like alpha particles or beta particles or Auger electrons. Like what are the isotopes it emit? The alpha particles mean astatine 211, like radium 223, bismuth 213, actinium 225. These are alpha emitting isotopes. It gives energy of 80 kilo electron volt per micromillimeter of tissue. This is called high linear energy transfer to the tissue. That's why it breaks DNA in high intense way. That's why once the damage is happened, the DNA, it cannot be repairable. Like how does it work? Like due to the alpha particle, it causes the damage on DNA. Again, indirectly, it during like when the isotopes in human body, it ionizes the water and produce the radioactive species and radioactive the cancer cells. Like other example of beta particle emitting isotopes like copper 67, yttrium 90, iodine 131, and lutetium 177. These are low energy linear energy transfer. That is, we call it as a low energy because it gives the energy of 0.222 kilo electron volt per micromillimeter of tissues. Like Auger electron is linear energy transfer. It emits 4 to 20 micromillimeter of tissue. Again, this kills the cancer very selectively due to the conjugating vectors. Due to vector, it reaches the targets and kill the only cancer cell. It doesn't harm the normal human cells. Like this slide highlights the beauty of the therapy. Like this patient is suffering on end-terminal cancer. He is suffering on stage 4 of the cancer. The cancer type is prostate metastatic cancer. Like again, they treated this, injected this patient with the gallium 68 with the PSMA. Again, this is a target-specific reagent. They injected the patient. That's why they identify the patient, what kind of cancer he is suffering. That's why we come up, the radiologists come up with the specific treatment plan for this patient. For this case, they injected actinium-225 labeled with the PSMA. After 3 doses, that the patient completely remains, completely cured from the tumor. See that prostate specific antigen level, like this patient before treatment, he had microgram per ml of antigen level. This is not good for the normal level. It should be 0.1 nanogram per ml. If it's higher, it's causing the patient. That's why he reached the end-terminal cancer. Like after 1 dose, he became very normal. This patient can live life at least 10 years without any cancer relapse. Like, as I said, we are aimed to develop target radiopharmaceutical agent development. For this purpose, we need to have three components. One is radioactivity or radioisotopes. As I discussed several examples like imaging, I said gallium 64 or zirconium 8. Again, we need to have linker or chelator to attach to the radioisotopes and link the targeting molecules. That's why it bind with the target and selectively they kill the cancer cell without harming normal healthy tissues or normal cells. Like, what going to be a vector? Vector, it may be small molecules or peptide or monoclonal antibodies or fragments of monoclonal antibodies. It serves as a vehicle to carry the radionuclide to the target. This previous slide. That's why we can label and inject the patient. We can get the meaningful data and treatment from out of it. Again, my current studies, we plan to use antibody. That's why I plan to, I thought it's good idea to discuss what is antibody. Antibody is a immunoglobulin, is large Y-shaped protein. It's mainly produced from plasma cell. When antigen like a bacteria or virus entered the human body, our body systematically antibody. This antibody has a unique functionality called fragment antigen binding. It gives it binds to the antigen very selectively and very exactly to the antigen. That's why it kills that antigen and cure the disease when antibody produced in our body. Like, as I said, the chelators are linkers. It's for the antibody based radiopharmaceutical, we need to have bifunctional chelator. Bifunctional means it should have both bioconjugating groups as well as radiometal chelating groups in a single molecule. Bioconjugating groups usually carries the reactive functional group. It forms different bond formation with the protein. Like, usually the protein has free amino functional. In antibody case, there are several free lysine. It reacts with the free lysine of amino functional and form different amide or thiourea or thioester bond formation. Depends upon the functionality. If the reactive group has active ester, it forms amide bond. If reactive groups has isocyanide, it forms thiourea reacting with the amine. If reactive groups has malamide, it forms thioester. Like, whatever I discussed on previous slide, this slide summarized all the things here. Like, as I said, this is the bifunctional chelator. It has the reactive functional group of thiocyanate. It reacts with the amine. As every antibody has at least 7 to 8 free lysine, it reacts randomly to free lysine and form thiourea. Once the bioconjugate ready, we can label with whatever therapy we plan or diagnostic study we plan. Suppose if you plan SPET imaging tracer, we have to label with indium. If you plan to do PET image, you have to label with the zirconium 89 or copper 64. Like, if you want to develop the same conjugate for therapy, you can label with the lutetium 177 for beta emitting, for alpha emitting actinium 225, or any Auger electron isotopes like iodine 121 or technetium 99M, whatever isotope you want. Like, for these studies, we decided to work with an insulin like growth factor 1 receptor. It's called IGF-1R. Like, most of the this is a transmembrane receptor. It's expressed mostly in human cancer, including 50% of triple negative breast carcinoma. Like, IGFR is the new target for the treatment of triple negative breast cancer. The patient express the IGFR. If you treat with the anti-IGFR, they may get benefit from this antibody. There are 8 anti-IGF-1R antibodies are entered to the clinical trial. Based on this one, we have selected as a cystosumab as our antibody agent. Again, these antibodies have high specificity to the IGF-1 receptor and it internalize and degrade the IGF-1 receptors and does not bind to the insulin-1 receptor. Also, it downregulate IGF-1R and the insulin receptor. That's why it decreases the cancer progression. Like, as I mentioned, we selected this study for cistuzumab. Cistuzumab is fully humanized monoclonal antibody. It, as I said, we can treat with the patient with the the patient is expressing breast cancer lesion expressing IGF-1 receptor. If we treat the same patient with the cistuzumab, they will get most of it benefit from this treatment. Again, in the literature, it demonstrated that it has anti-cancer activity in vitro and in vivo studies. Again, when we did the combination studies with another chemotherapy or radiotherapy, they get beneficial effect, like based on the synergetic and addictive effect. For this study, we want to develop pet tracer development. That's why we decided to label with we decided to work with the indium 111. Indium 111 is placed on the periodic table on a group 13. It has hard acid on the it nature of hard acid. It has a pK of 4. That way, it forms strong complex with the stable metal complex with donor atoms that have hard donor atoms. Like in the literature, it's reported that indium 111 form complex with the various bifunctional chelators called DTPA or DOTA or NOTA. Again, it's well-known PET tracer, well-known PET radiometal. It emits gamma proton at 2 energy level. That's why we can quantify the 2 energy and we can visualize the images. Again, it has half-life of 68 hours. It matches with the half-life of our antibodies called cistuzumab. It also has half-life is 3 to 4. Like we have to select the radioisotope in such a way that we have to match with our vector. It's suppose if our vector half-life is short, we have to choose radioisotope with the lesser half-life. If you choose with the radioisotope with the higher half-life, we cannot get most benefit from the isotopes. Like the first step of the project begins with the bioconjugation. Bioconjugation means we introduce this bifunctional chelator to the protein. For this purpose, I took 1 mole of cistuzumab and 16-fold of SS of DOTA thiocyanate. As I mentioned earlier, this cistuzumab has 8 free lysine aminofunctional group. It forms with thiourea bond formation. We want to have at least 3 DOTA molecule attached on this protein. Like if you attach 3 DOTA molecule attached on the protein, we can get good radio labeling. If you have few number of this DOTA molecule, the radio labeling may be not efficient. It cannot useful for the radio labeling studies. That's why we aim to have 3 to 4 DOTA molecule in the antibody. For this purpose, we try to incubate the protein and this chelator in basic pH for 2 hours on certain period of time. After that, we purify the sample with the help of centrifugal filters. It works. How does it work? It works based on the molecular size. Like whatever the higher molecular weight, as you know, the protein molecular weight is around 150K. Whatever the small, like I used the centrifugal filter of 10K, whatever the small molecules below 10K, it passes through the filter. Rest of them retain on the filters. That's why we can do centrifugal filter to purify this sample. Once the sample is purified once the bioconjugate is purified, we do the lot of QC because we expose this protein to strong basic pH for certain period of time. It may change. As we know, the protein is liable on acid and base. It may coagulate, it denature the protein. We want to make sure that the protein is intact and protein is pure. That's why we do several QC to check the protein purity and protein integrity by several QC or several assay. The first assay we did is bioanalyzer. For this, we simultaneously use a naked mean unconjugated antibody as well as conjugated or bioconjugated antibody. It clearly indicates that there is no other fragment bonds. It clearly indicates that the conjugated antibody is pure. It doesn't have any smaller molecule fragments. That's why we decided to do next QC test with the KD values didn't show any significant difference. We decided to do further QC test on real in vitro cell model. For we have selected some 1,4-PT9 cancer cell. This cell expresses the IGF-1 receptor. As I said, we have selected the antibody for anti-IGF-1R. That's why we want to select the cells based on our target. The some 1,4-PT9 express IGF-1 receptor. That's why we have, again, we did the cell binding studies. We didn't see any significant difference. We thought the bioconjugate didn't change the protein integrity or affinity behavior. We decided to go for next step. Like as I said, these proteins able to internalize the cancer cells. Again, we checked this protein able to internalize the cancer cells. We decided to the radio labeling. In radio labeling, like as you are aware of, and B to react to get C mean based on the molar. Like as I said, I took 1 mole of protein and 16 mole SS of chelated to get the protein conjugate. Here we cannot do the radioactivity such a way that for that purpose we use specific term is called specific activity. Like suppose if I take 1 microgram antibody, I have to use some specific amount of radioactivity. That's why we called specific activity. For optimization of this labeling condition, we use 4 different parameters. First one is specific activity and pH and incubation time and incubation temperature. Based on this, we found out the makes optimal reaction condition is that in if suppose if you take 20 microgram of our antibody, we have to take 10 megabecquerel of activity and pH 5.2 is the maximum to obtain a good radiochemical yield. I will discuss what is a radiochemical yield in next slide. Again, this kind of reacts, as I said, these are radio emitting or radionuclide or radioisotopes. It emits the energy. Like suppose like as a radiochemist, if I'm doing this kind of work, I may get unwanted dose. I don't want to get so much dose to do some routine lab studies. For that purpose, we use the shielded fume. The shielded fume mean these are fume is placed on the lead. Like lead is again on chemical element on metal. It protects the unwanted radiation. Again, for working these radioisotopes in Canada, we have very strict regulation covered by Canadian Nuclear Safety Commission. It's called the CNC. We have to use low amount of radioactivity as like how can minimize the unwanted radiation exposure to workers or other people in the lab. We have to work in the basic principle called ALARA, as low as reasonably achievable. That is ALARA. In the ALARA principle, we have to apply some parameters again. Like what are the parameters? We use time, field, distance. Like if the radiochemist want to do this kind of work, we limit the time. Again, we use the field like lead bricks or lead cave or lead field. Again, we make sure that proper distance to get unwanted radiation, avoid to unwanted get radiation exposure. Again, during the chemical reaction, in classical reaction, we check the reaction or check the reaction progress by ITLC or thin layer chromatography, like TLC, thin layer chromatography or HPLC. For this one, we can use the same principle, but we no need to elute the TLC with we have to elute the TLC. We no need to spray any reagent to visualize the spot. For that purpose, we use the radiometric detector. The radiometric detector detect where is the gamma photon energy is there. Like suppose if a molecule is like labeled, when you elute your TLC, it sticks on the bottom. It clearly indicates that you labeled your antibody conjugates or bioconjugates with your metal. That's why I can see the spot is staying on. Again, I see very single clear peak. I didn't see any free metal region. Like the free metal elutes on the solvent front. Like based on this ITLC, I can say I got the more than 95% of radiochemical incorporation onto my protein conjugate. Otherwise, it called as a radiochemical yield. Like if it have more than 95% of radiochemical yield, we no need to do the further purification. We can go, we can take this tracer for further studies. Again, as I said, we expose this protein conjugate and acidic pH. Like when we did the bioconjugation, we exposed protein in a basic pH. That's why we checked several QC to know the protein binding, affinity, and protein integrity and purity by several QC. Again, we do several QC. For this purpose, we use different kind of instrument or radiometric detector. Like again, I checked this labeled tracer with the HVLC. It has the radiometric detector. That's why we can see the where it's my protein labeled radioconjugates. This is radiometric channel. This 220 channel and 280 channel, it gives my protein peaks. This information about where it's my antibodies labeled. Like again, as I said, we exposed with this protein conjugate in a basic, acidic pH for radio labeling purpose. We have to check several QC to check the protein integrity and purity, affinity by several QC methods. First one, we do stability studies. Like we want to know this tracer should be stable. As you know, the radiometal, as I said earlier, the radioisotope, the half-life mean, like for indium, it has half-life of 68 hours. Like if I take 1 millicurie of activity, if I go and measure the same activity after 68 hours, I see only less amount. That is mean 50% is decay. All the isotopes decay and release the energy. We have 50% is left over. Again, the 50% decay, like the isotopes decay at certain period and became to reach the stable metals. Once if it forms the stable metals, we cannot get any radioactivity from that. We have to have this tracer should be stable during our animal studies as well as in vitro studies. That's why we want to check the stability of this product in a two different conditions. One is the formulation buffer in PBS, because we decided to do, like we do most of the animal studies in PBS. That's why we took PBS as a plasma, like because we going to inject the tracer into animal or human. It should be stable in a human or animal. It should be compatible with the plasma. That's the purpose we have chosen two different conditions. Again, this tracer was more stable and maintained the purity of more than 95% up to 5 days. This tracer was stable. That's why we decided to do some more QC on real in vitro cell model. The first studies we did this immunoreactivity fraction. Like immunoreactivity mean, again, we exposed with this protein conjugate. Each protein conjugate has own immunoreactive fraction. If you expose basic condition or acid condition, it immunoreactive fraction may be diminished. Like that's why we want to check the immunoreactivity fraction for this studies. What we did, we take such like fixed amount of radioactivity treated with the various range of cells and do some incubation and collect fraction and do some like washing and collect the supernatant liquid and collect the pellet and measure the activity with the help of this gamma counter. Like gamma counter measure the gamma photon based on the isotopes what we are using. It measures the gamma photon at very small amount, like even in terms of, if you want to say in terms of radioactivity in very low nanocurie level. Like we used to say activity in various unit or various units of activity. The high amount is called gigabecquerel, very low amount is called nanobecquerel or kilobecquerel or nanocurie. That's why this instrument able to detect on even very trace amount of activity in the cells. The another experiment we performed with that, it's cell binding assay. For that, take your pellet, measure with the help of this gamma counter, and plot with the Excel sheet, and we will get the KD values. Again, the KD values didn't change that much. That's why we decided to do animal studies. For first animal studies, we did with mice genograph model. For that purpose, we developed the genograph. How we develop the genograph mean we grow the cancer cells on the Petri plates, collect the cancer cell, and inject on the nude mice on subcutaneously. After 2 to 3 weeks, you may see the tumor where you injected your cells. For that purpose, we injected the cells with the matrigel. That's why it's stick on where you injected. Like if you injected without matrigel, the cells may be distribute to some other body, it may form some metastasis. In order to avoid that, we use the matrigel. Again, why we are using nude mice? There, if you use the normal mice, it has capacity to immune reaction to reject the tumor formation. Nude mice has capability, it don't have any capability to reject the tumor. That's why we prefer to use the generate the tumor model on the nude mice. Once the tumor model is generated, we inject the tracer. For these studies, we injected dose of 5 to 7 megabecquerel per microgram. In terms of protein concentration, the protein amount we injected less than 10 microgram. Again, for tailwind injection. Why we injected tailwind injection mean most of the radiopharmaceutical in clinical, they inject as a intravenous bubbles injection. Like intravenous injection. That's why we try to mimic the same condition. That's why if the preclinical moves to the clinical level, it may have the same reproducibility. Like once the tracer injected on the mice, after some time, like for this study, we collected organs after 72 hours, and we collected several organs. Again, with the help of gamma counter, we measure the amount of activity in various tissues, and we plotted the graphs with the help of graph prism. For the same study, we used human IgG. Human IgG, it doesn't have any IgG of 1R expression. This cystosome has IgF 1R expression. That's why we inoculated someone for PT cells. If your tracer has affinity to bind the IGF-1R, it should take on the tumor. If your tracer has any affinity on the tumor, the uptake may be less, or the uptake is maybe non-target uptake. That's why we see the clear difference between positive tracer and this negative tracer. The tracer uptake was very selective. That's why we decided to do SPECT imaging studies. For this SPECT imaging studies, we used this SPECT CT scanner. It's available on our facility. For this purpose, we selected again 2 groups, one group with the positive, one group with the negative. We injected the tracer about 10 megabecquerel, 2 times higher dose than BioD study, that much sensitive than gamma counter. That's why we injected the amount of protein and amount of activity slightly higher than BioD studies. And we adopted the same parameter for the image acquisition. And after image is accused, we do the reconstruction of these images. That's why we can visualize the mouse. See, like when, as I said, this is the cystosome injected mice. Again, the tumor has IGF-1 receptor. That's why there is a selective uptake on tumor. That's why we visualize where are the IGF-1 receptor. It's clearly indicate that it selectively accumulate on the tumor. The same mice group treated with the negative control, there is no tumor uptake. It clearly indicate that we can visualize the images wherever the IGF-1 receptor expression are there. Again, we do in-depth image analysis. We draw region of interest on the various organs and know where is the activities accumulated. Like again, you may see some uptake on activity on liver, some tracer, like some compound, some molecule or some vector tend to clear to the renal clear system, but antibody used to clear through the hepatopylori system. That's why we able to see some liver uptake and spleen uptake. Again, we did the in-depth image analysis. For that purpose, we draw the region of interest on various organs and found out how is the uptake in the organs. Like for this case, we did in-depth analysis on tumor and liver because liver is a clearing pathway. And again, we took as a muscle as a background and we calculated tumor muscle ratio. Like see, the uptake is taking maximum at peak at 48 hours. It starts clearing from the tumor. It's the same tendency in the liver too. Like you start picking up at 24 hours, you start clearing on after 120 hours. Like based on that, we calculated tumor muscle ratio. See, the tumor muscle ratio values are opposite than tumor. Like what does it indicate mean? Like when you correlate this tumor uptake to the background of muscle, we can visualize the uptake based on these images. When the later time point, you see that there is no uptake on the liver, only there is uptake on the tumor. Suppose if this tracer goes to the clinic, you no need to image the patient on 24 hours. You can image the patient up to third day to fifth day. No need, like it gives the convenience for the radiologist. Like some tracer, like if you inject the F18 tracer to your human body, you have to image the patient right away within 1 minute. But if this tracer enters to the clinic, we no need to scan the patient on the day of injection. You can wait. It gives the convenience for radiologists scan the patient and give some time to do the image analysis for the radiologist. It gives a convenience for the clinical purpose. In order to validate this concept, we took another xenograft model. Again, this that clear uptake in a tumor. There is no uptake on the negative tracer. It's clearly indicate that when you label with the IGF-1 or antibody, we can visualize the tumor and its uptake is very selective on the tumor. Again, we did in-depth analysis. The trend is more or less same with some 1,4-PT xenograft model. The trend are more or less same. Again, it's peaking at 48 hours. It's the end of the last imaging studies, like we did biopsy studies. See the clear-cut difference. The tumor uptake is very selective compared to the negative control of human IgG. That's why we decided to label with this tracer. Like whatever the bioconjugate we labeled with the indium, we can use the same bioconjugate and label with the actinium. That's why we call it as a thornostic. Like the same tracer can be used for diagnostic purpose. For this case, expressing IGF-1 receptor, they can diagnose with the help of indium 111 labeled tracer. If you want to treat the same patient, you can label another alpha emitting isotopes and inject this tracer to the patient, and you can see the result. What I shown in the clinical data of actinium-225 with the PSMA. Why we selected actinium-225 for this studies mean it has half-life of 10 days. Like during emit alpha particles, it has wide variety of energy, like it gives 5.8 to 8.4 milli electron volt. Again, the TC range is like very selective. It was causing selective damage on the RNA. Once the damage is happened, the RNA never going to repairable. Again, the cancer cell death is, we cannot kill any non-cancer cells or healthy human cells. Again, for these studies, the same protein conjugate we labeled with the actinium 225. For this, again, I played with the four parameters: specific activity and pH and incubation time and temperature. We found that this is the specific activities maximum to get the maximum radiochemical yield of more than 95%. For these studies, again, we did all the QC studies, what I mentioned for the indium tracer. Like we did the stability studies on PBS and stability studies on serum. Again, we did the cell binding affinity and immunoreactivity fraction assay. Once those results are clear or those results don't have any red flags, that's why we decided to do the therapy studies with the same tracer. This therapy, we take 5 different groups. For the first group, we took cistazimab. As I said, cistazimab itself has anti-cancer activity. We want to test that does it have real anti-cancer activity on the same model. That's why we selected as a positive control. Again, we choose 2 different specific activity of actinium labeled tracer. This is the tracer we injected 0.5 milligram per kilogram dose. The another group we injected 2.5 milligram of kilogram of dose. Again, we took control tracer and one PBS treater, like mean just PBS group, no radioactivity or no positive substance. Like again, for each group, we had 6 to 8 mice, and we once the xenograft is developed and it's reached, like if you saw the tumor on the mice, like once if it reached the 20 millimeters cube, we start the treatment. We gave 2 different doses, like one dose on day 10, the second dose on day 20. See the real in vivo effect. Like this group, like 2 mice is completely relapsed the tumor. Like when the mouse is untreated, all the tumors dead before 90 days. Based on this data, we measure the mean survival time, like survival graph. It clearly indicates that beauty of this tracer for the therapy. Like when the PBS treated, the mice is surviving, like 6 mice was survived up to 52 days. Like the same days when the dose was given with low specific dose of 2.5 mg per kg, it served 120 days. The difference is significant. Like it's amazing result, we can say. Yeah, that's it. Like we able to make good protein conjugates and be able to label with the indium 111 and actinium 225 with the high radiochemical yield. Again, we able to see the selective accumulation on the tumor. Again, where why we saw the tumor mean the tumor are expressing IGF-1 receptor. That's why you're able to see the visualize the IGF-1 expression. Again, when we treated this tracer on the mice with high dose of labeled tracer of actinium 225, 2 mice was completely relaxed the tumor, like completely cured and served more than 180 days. When the mice was untreated, all the mice were dead at before 52 days. This is amazing result. We published this result on some high impact factor journal called Molecular Pharmaceutics, impact factor of 5. The another work we published in scientific report, impact factor of 5. On team effort, we cannot achieve this kind of result. Again, I would like to thank our funding agency and from like facility provider from Frederick Center. That's it. Thank you for your kind attention. If you have any question, please feel free to ask me. Thank you.
Friends, if you want to ask any question, okay. I would like to request Yarini, third biochemistry, to propose the vote of thanks.
Good morning. I, Yarni M. from 3rd BSc Biochemistry, on behalf of the Department of Biochemistry and the entire fraternity of the institution, take it as an honor to propose the vote of thanks. First of all, I extend my most sincere thanks to the Almighty God for making today's event a resounding success. With His blessings and grace, we can make this event what it was. I give a hearty thanks to our chief guest, Dr. V. Raja Solomon, who spared time from his busiest schedule to cherish the occasion. Today, we had an opportunity to hear your thoughts, and this will surely be going to encourage us in our future events. Your thoughts have enlightened our mind and have shown us a new path. My gratitude to you, sir, for your presence in the occasion and sharing your knowledge. A very special mention to our secretary, Dr. Sister Mary Josephine Rani, Principal Dr. Sister Jayashanti, Vice Principal Dr. Sister Amala Valamati, Shift 1, and Sister Juliana Agnes Victor, Shift 2. Our ever-loving HOD ma'am and professors for being the catalyst that stimulated us to do our best and stand as pillars of strength. Finally, I would like to thank all of you joined here for making the event a grand success. Thank you one and all.
Thank you, Yarini. Thank you, sir. Thank you for taking your time among your busy schedule. Thank you, sir.
Yeah. It's my pleasure. Thanks for having me. Thank you so much. Bye.
Thank you, sir. Thank you.